So guys, let us continue to our lecture 2 on surface phenomena. So at the end of this lecture, students are expected to understand the surface tension and pressure difference, which give rise to a Young Laplace equation. And you are expected to understand and able to explain conceptually on how to measure surface tension, and understand and able to calculate surface tension, as well as to calculate the work of adhesion and work of cohesion. So another way of expressing surface tension is in terms of pressure difference that exists across a curved surface. For example, when we look at a bubble, okay, and inside this bubble you have a pressure 1 and outside the bubble is pressure 2, and at equilibrium the bubble remain as a sphere, which there is a pressure difference, which is also a function of surface tension. So let us consider a bubble with a radius of r, okay, here, and let's say when this bubble contract, okay, to a smaller size of bubble, okay, with a work of distance of dr, okay, what is the work done for this contraction to happen? Okay, as you know, the contraction is caused by surface tension of the film, okay, and when the bubble resists as a bubble, it is a result of internal pressure of the bubble. So meaning that you have a certain pressure internally that they can resist the bubble to collapse. Okay, so the work done to change the bubble from a radius of r to a new smaller bubble is equals to surface tension times the changes of area involving in the sphere of this bubble minus the energy that caused by the pressure, which is PV, the change of pressure with the volume of the initial volume and the final volume of the bubble. So how we can find dA? So as you know, the surface area of a sphere is 4 pi r square. So dA is equals to calculus of 4 pi r square. Okay, so perform the calculation, it will be equals to 8 pi r dr. So how about V? How you know the volume of a bubble? So the volume of the bubble is similar to the volume of a sphere, which is 4 over 3 pi r cube. Again, perform calculus and you get 4 pi r square dr. Okay, so however, after the work done, okay, at equilibrium the contraction force of this bubble must be balanced up by the pressure forces, which means gamma dA must be equals to changes of pressure dV. Okay, take the equation here to replace dA and equation here to replace dV. Okay, then cross out whatever is similar and you got delta P is equals to 2 gamma R, and this is what you call the Young Laplace equation that relates surface tension to pressure difference.
How surface tension relates to physiology, it's important to describe something called the law of Laplace. Imagine a hollow sphere made of some form of pliant material like plastic, for example, and inside is a thin layer of water coating the inner surface. The scale of this thin layer is greatly exaggerated in this picture, but the layer of water is made up of individual water molecules, all of which are attracting each other. The net sum of the circumferential attraction is that the water is actually being pulled inward. If there was no enclosed gas, the water would contract down into a ball. However, if there is enclosed gas, the result is an increase in the pressure of that gas. The surface tension pulls the water inward until an equilibrium is reached with the pressure pushing outward. The law of Laplace relates the surface tension to the enclosed pressure. It states that the pressure inside the spherical bubble is equal to 2 times the tension divided by the radius. Let's take a look in the lungs to see how this is significant. If we zoom way in, we will see that the two lungs are actually composed of roughly 1 billion sacs of air called alveoli, which are connected via an intricate branching network of air passages. Most of the details of this are not relevant for this discussion, other than to say that the alveoli are the site in the lungs where gas exchange occurs, and it's critically important that they stay open and don't collapse, or else oxygen will not be able to make its way into the bloodstream. So how is the law of Laplace applicable here? First, the lungs are a very moist place, and the insides of the alveoli are coated with water. Some alveoli may start off larger than others. Since the surface tension of water is independent of the size of the alveoli, based on the law of Laplace, a large radius results in a relatively low pressure, while a small radius results in a relatively high pressure. So when a patient takes a breath of air in, the air travels preferentially to the large alveolus with low pressure. Not only that, air could even travel backwards from the small alveolus with high pressure. The consequence of this is that large alveoli risk becoming overdistended, and small alveoli risk collapsing altogether, a condition known as atelectasis. Alveoli that have totally collapsed can no longer take part in gas exchange, leading to diminished ability of the body to take in oxygen. This is obviously not good, so the body must have a way to prevent this from happening. It does, and it involves something called surfactant. About pulmonary surfactant, which is a complex lipoprotein formed by cells called type 2 pneumocytes, which line the alveoli. The most notable constituent of surfactant is called DPPC, or more formally, dipalmitoyl phosphatidylcholine. This compound greatly reduces surface tension of the fluid coating the inside of the alveoli. Interestingly, unlike water, the surface tension of the mix of water and pulmonary surfactant is roughly proportional to the surface area over which it is spread. In other words, the power of surfactant to lower surface tension is most pronounced when the alveoli are smaller than when they are distended. Here are our two alveoli again, but the smaller one no longer has a relatively high internal pressure compared to the larger one. Instead, surfactant lowers the surface tension in the large alveolus by a modest amount, while it lowers the surface tension in the small alveolus by quite a bit. The fact that the surface tension in the small alveolus is particularly low offsets the effect of the smaller radius. As a consequence, the resulting internal pressure is low within both alveoli. So when air comes into the airways, both alveoli are inflated, improving gas exchange. So what are the net consequences of pulmonary surfactant? First, a reduction in the collapse of underinflated alveoli, otherwise known as atelectasis. Second, a more even distribution of ventilation amongst the alveoli, improving oxygenation. Third, it improves lung compliance. For the most part, a discussion of lung compliance falls outside the scope of this video. However, very briefly, lung compliance is a measure of how much the volume of the lungs change in response to a change in pressure. The lower the compliance, the greater the pressure difference that must be generated in order to exchange the same volume of air. A major contributing factor to lung compliance is the alveolar surface tension, which has a cumulative tendency to pull the lung in on itself and resist inflation. Therefore, surfactant increases lung compliance, reducing the amount of work a patient or ventilator must do during inhalation. The last major clinically relevant aspect of surfactant has to do with preterm births. In humans, surfactant is not reliably produced until about 34 weeks gestation. Therefore, premature infants born prior to this may have significant difficulty with oxygenation and ventilation, a condition called infant respiratory distress syndrome, not to be confused with adult respiratory distress syndrome, which is quite different. Treatment of infant respiratory distress syndrome consists of a combination of positive pressure ventilation and exogenous surfactant delivered to the alveoli via instillation into the airways while intubated. If a mother is suspected to be at high risk of preterm delivery, she can be given antenatal corticosteroids, which speeds up the infant's synthesis and release of surfactant.
So after we have understand the relationship between surface tension and pressure difference, as well as the definitions of a surface tension and surface free energy, let us go into the methods for measuring surface tension and interfacial tensions. There are 4 different methods can be used, which is the capillary rise method, ring tensiometer, Wilhelmy Plate method, and drop weight method. So first, let us look at the capillary rise method. The principle of capillary rise method is based on the capillary actions. So in this method, capillary tube is placed in a liquid. Then the liquid will rise to a certain height in the tube. For example, what happened from here to here. Okay, and the principle is the interaction force of the liquid with the capillary wall, where adhesion are stronger than those between the liquid molecule. Thus, the liquid wet the wall well and rises. Okay, at the maximum height, which is here, a maximum height, the opposing force are in equilibrium. What do you mean by opposing force? So by capillary action, they will go up. So the opposing force will be the one with gravity. Okay, so if the tube is small enough, the meniscus can be considered as hemispherical. So here is the hemispherical shape for ease of calculation. Okay, so here are some examples of the capillary actions. So for example, you have a very tiny tube, and the solution will be drawn by this very tiny glass rod, and also it will be drawn up by a tissue. Okay, this is what you call capillary actions. And when you have 2 slides together and a very tiny space between the slides, can actually draw the liquid by, again, the same capillary actions. It is mainly because that the adhesion force between the water molecule and the wall of the slide is higher than cohesion between the water molecule. So as I have mentioned just now, when the liquid of the capillary rise to a certain height, which is what you call a maximum height, okay, the force upward is equals to the force downward. Okay, and consider the surface of the meniscus as a hemisphere here. Hemisphere. Okay, what is the force upward? Okay, and if this is hemisphere, so the force acting at this direction is what you call a surface tension. Okay, so to consider the vector, okay, for example, here is your angle of the meniscus to the wall. Okay, to perform vector to this surface, to make it upward, so it will be equals to gamma cos theta, right? So the force upward equals to gamma cos theta times the perimeter of this meniscus, which is equals to 2 pi a. In this case, a is the radius. Okay, so what is the force downward? So force downward, all right, is equals to PVG, right? So P is the density, okay, which is known, okay. G is gravity. So what is V? Consider the whole capillary. For example, you have a capillary here that they rise up to here from this what you call from this container. They rise up to a certain height, h. So here, from here to here is what you call a cylinder, right? And what is the volume of a cylinder? So volume of a cylinder will be the area of this hemisphere times the height. Okay, what are the area of a circle? Will be pi a square times the height, okay, and g. So the force upward is equals to the force downward at equilibrium. Okay, so in that case will be gamma 2 pi a cos theta equals to pg pi a squared h. Alright, so just cross out the pi, cross out the radius. So what left over will be, so gamma will be equals to pi g a h, okay, divide by 2 cos theta. Okay, so following from the previous slide, when we have sought out that the gamma is equals to, okay, density gravity ah over 2 cos theta, and when a liquid wets the glass completely, which happened in the very thin capillary, so we assume theta is equals to 0. So cos theta will be equals to 1, and you will finally have gamma equals to half of density, gravity, radius plus height. Okay, so using this experiment, for example, let's say you know the height and you know the radius of the capillary, that rod that you use, then you can easily calculate the surface tension of the liquid tested. So let's try this example. A sample of chloroform rose to a height of 3.67 cm at 20 degrees in a capillary tube having an inside radius of 0.01 cm. And what is the surface tension of chloroform at this temperature, given that the density of the chloroform is 1.476? So using the derived equation, you can easily substitute the radius 0.01 cm, substitute the height, which is 3.67 cm, substitute the gravity, okay, and also the density, which is 1.476 gram per per cm cube. So looking at all these units, you have to bear in mind that the unit that you use for the gravity must be equivalent. Alright, please try it out. Okay, so this slide shows the answer, which is 0.01 cm, 3.76. Just substitute with 981 as the equivalent units. Then you can have the surface tension of 26.6 dyne per cm. So the capillary rise method is considered to be the most accurate method in measuring surface tension, as the surface of the liquid measure is undisturbed throughout the measurement. However, it is used a little in pharmaceutical research, and also it is not possible to obtain interfacial tension using this method. We can only measure the surface tension of a pure liquid, but not the interfacial tension. So the second method that you can use to measure surface tension is ring tensiometer. So the principle of this method is that the force necessary to detach a platinum indium ring immersed at the surface or interface is proportional to the surface or interfacial tension, and the force of detachment is recorded in dyne on a calibrated dial. So here is your calibrated dial, okay, and you immerse the ring here. Here is a ring in your liquid, and you try to increase the weight from this dial, okay. Then they will record the force to break the ring detach from your liquid. This is what actually you perform in our FAR 221 surface phenomena experiment. Okay, here is some of the example that you can see.
The ring probe is immersed in the liquid sample. As the ring is raised, a meniscus forms, pulling down on the ring. The force exerted on the meniscus is recorded and can be used to calculate surface tension. Results are plotted and analysed by the attention software.
So by having the detachment force, how we can actually measure the surface tension? So the surface tension is equals to the reading in dyne, okay, divide by the 2 times of the ring circumference. Alright, why is it 2 times of the ring circumference? If you look at this diagram, okay, here is your ring, okay, and here is a cross-sectional diagram of this ring. So when they are trying to detach from the ring, what happened to this ring? So you have a ring here, right? They are trying to detach. Actually, there is 2 surface detachment from one ring. Okay, so that's why you have to times 2, because here 1 and behind another 1. So you have to times 2 and ring of the circumference. So the ring of the circumference will be just the perimeter of a circle, which is 2 pi r. Okay. So the third method that you can use is Wilhelmy Plate method, and the principle of this method is the apparatus consists of a thin glass or platinum plate attached to a suitable balance. So you attach to a balance, okay. The plate is immersed in the liquid, and the liquid container is gradually lowered down. So let's say here is a container, and the container is gradually lowered down, and the reading on the balance is recorded immediately prior to detachment. So the principle is quite similar to the ring method. It's just that the difference is what they use is a plate instead of a ring. So here is the video of how they use the plate method to determine surface tension experimentally. So that you can see they wet the plate first before they actually make the measurement. Then they make the plate immerse into the liquid. Then they start the measurement by slowly immerse fully the plate onto the liquid, and then lower down the stage of the liquid and calculate the detachment. So now they start to lower down the passage. Very slowly they lower down the liquid. Yeah, now the force is being recorded. So mathematically, to calculate the surface tension, the detachment force is equals to the surface tension multiplied by the perimeter of the surface of detachment. It is just similar to that of the ring. The detachment of that in the ring method is multiplied by the perimeter of the ring, and here this time it is multiplied by the perimeter of the surface detached. So which is, they have the what you call here L, and you have T. Okay, so you have surface of L and T are the length and the thickness of the plate. So here is your plate, okay, with the length which is L and the thickness of the plate. Then you can calculate the surface tension. And what is WL? WL is the reading on the balance before detachment. You have to minus the weight of the plate, which is the second term. So the last method that you can use to measure surface tension of your liquid will be drop weight and drop volume methods. So the principle of these methods is that if the volume or weight of a drop as it is detached from a tip of known radius is determined, then the surface tension can be calculated using this equation, whereby the m will be the mass of the drop, v will be the volume, okay, g will be the gravity, and r will be the radius of the tip. Okay, so in the subsequent slide it will show you experimentally how we can actually measure surface tension using this drop weight and drop volume method. So this is the example of a machine that we use for drop weight methods.
A drop is first dispensed from the needle. This technique can also be used to measure the interfacial tension between two liquids. You can also visualize the pennant drop on your PC. The drop images are first recorded by the software for analysis. Then, the red baseline is used to mark the tip of the needle. The blue box determines the area where the drop shape can be found. Then, the drop shape are fitted using the Young-Laplace equation. The fitted curve can be seen as a border around the drop. This graph shows the surface tension measurements and average at known STS concentration. This measurement is performed on 20 STS solutions of different concentrations.
So now you know that how drop weight and drop volume method are carried out in the lab, and this method is easily adapted for both surface and interfacial tension, therefore it is very popular. However, a correction factor is required as not all the drop can leave the tip on detachment, and these correction factors depend on the radius of tip as well as the drop volume. It is also very important that the tip is completely wetted by the liquid before the experiment so that the drop does not climb up outside the tube. So just to make sure that the detachment is complete. And the drop should also be formed slowly, especially in the stage immediately preceding the detachment, as shown in the previous video. So now, just to recap the method that we have learned just now. The first one will be the capillary rise method. The second one is a ring tensiometer. The third one is the Wilhelmy Plate method. And the fourth one is drop, weight, and volume methods. So now, which method to choose? So the choice of the method for measuring surface as well as interfacial tension depend on whether the surface or the interfacial tension is to be determined. It is because if you just determine the surface tension, the most accurate you can use capillary rise. However, when you want to determine interfacial tension, this method cannot be used. You may want to go further into the second, third order final methods, and the accuracy and convenience desired. Okay, for example, when you have your country very warm, and upon your drop, for example, we talk about the drop weight method, you have a tip, and upon your drop is formed, there will be evaporation. Then it is not accurate. Okay, in that case, you need to control the environment for your experiment. All right, and also the size of sample available. Okay, if you have a bigger size of sample, for example, you have 200 ml, 300 ml, you may actually go to the second and the third method. Okay, however, when you have a very little sample, you may want to try the drop weight and drop volume methods. So the last factor determining which method to choose is whether the effects of time on surface tension is to be studied. Okay, it is when we are trying to study, for example, wetting of a solid surface, okay, over time. Then the drop weight method will be used. Okay, for example, you have a tip here, all right, and this is your drop, and this is the surface to be wetted, all right, and after the detachment here, so you have your surface with the drop. Okay, so here will be the interfacial tension, and it will change over time. Okay, because there might be some some portion of the liquid absorbed into the solid which change the initial and final value of the contact angle. So question, what happened to the surface tension when temperature of a solution increased? Okay, first you have to think about what is the source of surface tension. It is intermolecular forces. Okay, and what happened to these forces when the temperature of a solution increase? Okay, when we have the forces of the same molecule, we call it cohesion force, right? When we have force for different molecule, for example, this one bind to this and bind to a different one, this is a adhesion forces. Okay, so what happened to the cohesion and adhesion forces when temperature of a solution increased? So in the molecular level, when a solution increase, okay, if you still remember your either form 6 or matriculation, what is the effect of temperature? Do you still remember the Boltzmann temperature curve? You have your kinetic of your molecule at this temperature, lower temperature, and it will move to the kinetic curve will move to the higher energy level when temperature increase. Okay, so the answer will be the surface tension of most liquid decrease almost linearly with an increase in temperature. That is, with an increase in kinetic energy of the molecule. Because of the kinetic energy of the molecule, the cohesion between the molecule is reduced. So surface tension reduced. Alright. So here are some example of the surface tension of water. When it is 0 degree, it is 75.6, and 20 degree, it is slightly lower, and when it is higher temperature, it has 63.5 surface tension. Therefore, it is necessary to control the temperature of system when carrying out surface and interfacial tension determination. So after we have understand surface tension and interfacial tension, let us go into the molecular level when we talk about the adhesion and cohesion. So the work of adhesion, which is the energy required to break the attraction between the unlike molecule. So surface or interfacial work is equals to the surface tension multiplied by the area of an increment. Okay, so work is equals to surface tension times the change of surface area. Okay, so hence the work required to separate two sections of liquid, each with a cross sectional of 1 cm square, is equals to the surface or interfacial tension multiplied by the area of increment. So we have example in the next slide. So now says you have this liquid and this is solid. Okay, so you have here is your surface tension of liquid and here will be your surface tension of solid, and the one that connect between liquid and solid will be surface tension of solid and liquid. Okay, what happened to the work of adhesion? Okay. The work of adhesion is the work done per unit area produced. So which mean, so from this big cylinder and you break into two, so what are the areas generated? So it will be the one here, bottom here, and this area. Okay, so the work done is the work done per unit area produced. So it is simply equals to the new area, this one and this one, alright, plus surface tension of solids minus the initial area. Okay, that will be the generated new area, right? So it will be the work of adhesion. Okay, so what is the work of cohesion? So when we have a single matter, for example, here is the liquid. Okay, so you generate the new area, which is here, as well as the one here. Okay, so what you have, the work of cohesion is the work done per unit area produced. Okay, when you have a single liquid, there is no initial interface, right? So in that case, the work done per unit, okay, the work of cohesion is equals to the two newly generated area, which is this one and this one. Apparently in this case, two newly generated is surface tension of liquid plus surface tension of liquid. It is equals to 2 surface tension of liquid. So cohesion and adhesion. Then you will have the terms spreading. So spreading occur if the work of adhesion is greater than the work of cohesion. So cohesion, for example, when you are talking about, you are trying to spread. Okay, this is a solid and this is a drop of liquid. Okay, you are trying to spread the liquid on top of this solid. Okay, so spreading occurs if work of adhesion is greater than the work of cohesion. So meaning that the attraction between the solid and liquid should be higher than cohesion. Okay, so spreading coefficient is equals to WA minus WC. Okay, so when this liquid can wet the solid, your spreading coefficient should be a positive. Okay, when it can't wet the solid, then it will be negative. Okay, so S can also be expressed. Okay, if you still remember when we just now we have the work of adhesion and work of cohesion equals to 2 gamma liquid and equals to gamma solids plus gamma liquids minus gamma solid liquid, right? So you just substitute WA and WC into this spreading coefficient equation, then you will get the final formula, which is S is equals to surface tension of solid minus surface tension liquid plus surface tension of solid liquid. Okay, so in this case, S is the surface tension of the sublayer liquid and L is the surface tension of the spreading liquid. So in this case, the sublayer liquid is referring to this solid. Okay, and the WL is the interfacial tension between the two items. So when you talk about the two liquid, then it will be interfacial tension between the two liquid. If you talk about the liquid and solid, then it will be interfacial tension between the liquid and solid. So looking further into spreading coefficients, so let's say look at the case of a drop of liquid on top of another liquid. Okay, so when this liquid A is dropped onto liquid B, what is the spreading coefficient? Okay, and however, once it touch liquid B, it might have a certain value of coefficient, which is the initial coefficient. Okay, And if you leave it on top of this liquid B after equilibrium, it will have what you call a final spreading coefficient after equilibrium. It might not be the same as the initial. Okay, so before equilibrium is reached, however, the water becomes saturated with the spreading material, which in turn becomes saturated with water after some time. So if we use a prime to denote the flowing equilibrium, which is the final value, then the surface tension are, for example, the surface tension of S prime and surface tension of L prime. So when mutual saturation has taken place, means after equilibrium, the spreading coefficient may be reduced or even become negative. This means although initially spreading is positive, okay, so means spreading may occur on the liquid, and it can be followed by coalescence of the excess material into a lens if the final spreading coefficient, which is the S prime, become negative in value. And this reversal of spreading take place when the surface tension of the S become less than the surface tension of the interfacial and the surface tension of the initial liquid. Okay, and the value of the interfacial tension does not change because the interfacial tension is determined under condition of mutual saturation. So in that case, only thing that can change is the surface tension of the pure material, okay, before and after equilibrium. However, interfacial tension is not changed. Okay, so example for question. So if surface tension of water is 72.8, okay, please remember this figure at 20 degree, and the surface tension of benzene is 28.9, and the interfacial tension between them is 35. And what is the initial and final spreading coefficient if after a certain time the surface tension changed to 62.2, and however for the benzene become 28.8. Okay, so let us write down everything in a mathematical language, which is S is equals to 72.8, okay, L is equals to 28.9, okay, and interfacial tension SL is equals to 35, okay, and gamma S prime is equals to 62.2, and gamma L prime is equals to 28.8. Okay, and as you know, spreading coefficient that we derived the formula before is equals to gamma S minus L plus LS. So you have gamma S 72.8, okay, minus this plus this. So this is the answer, 72.8 minus this and this, and you have the initial spreading happen. Okay, and however, because after the equilibrium we have the final surface tension 62.2 and 28.8. So what is the spreading coefficient after equilibrium, which is this is the answer, and it becomes negative. So therefore, although benzene spread initially on water, however at equilibrium there is formed a saturated monolayer with the excess benzene forming a lens. Means, okay, let's say this is a surface of water, so the benzene will actually form a lens on top of the water instead of spreading throughout the surface of water, but it forms a lens. Okay, So for extra note, in case of organic liquid spread on water, it is found that although the initial spreading may be positive, however, the final spreading coefficient always has a negative value with a monolayer form on top of this water surface. That's why it is important to consider the type of molecular structure that lead to high spreading coefficient. For example, when you have oil actually spread over water because it contains a polar group such as COOH or OH. So if you still remember the rules of light to light, so this polar group can increase the adhesion between oil and water. And as the carbon change of acid, oleic acid, for example, increase, the ratio of non-polar to polar character decreases and the spreading coefficient on water decreases. So many non-polar substances such as liquid petroleum, they fail to spread on water because they have the spreading coefficient of negative 13.4. Okay, benzene spread on water not because it is polar, but because the cohesive force between its molecules are much weaker than the adhesion for the water. So with that, I end my lecture on the methods for measuring surface tension and interfacial tension.